everyone, this is Marwa from Drug Marvel. Today we're going to explore how to identify sites on the protein that are specific to that protein among its homologs using a tool called BCSpecificitySearch. First of all, the BCSpecificitySearch belongs to a group of tools called the BCSearch. So let's start by typing that here in Google and know what is that. BCSearch, yes, that is it. This is the paper. I advise you to read it to get a better understanding about it. That is the BCSearch server. We will click on that. Now we are going to see this server. It tells you the review, it tells you what is it and why they have developed these tools. Actually, this is the summary of the BCSearch services. This image is actually from their paper, and I advise you to go through that. I put the link in the description box below, and here is it. They say that you have a query structure or fragment. This is your query protein, and you have a database, what they call the search database or the search space. Those are the structures to mine, and they have developed 4 tools. The first tool is called BCFragSearch. It is more concerned with certain fragment conformations. The BC mirror search is to search for fragments that are mirrored conformations for each other. And the BC loop search is concerned more with loops. It looks for the conformations matching loop length geometry. And what we are more concerned with in this tutorial is the BCSpecificitySearch. It identifies regions of protein with the specific conformations. So those are the 4 tools, and they explain it for you. I advise you to go through that and read it to know how they calculate the score and what are they. And let's read that one. This is the score. In practice, the score is SP. It's calculated as 1 minus n hits over n total, where n hits is the number of proteins where a similar fragment was found, and n total is the total number of proteins in the search bank. Thus, SB varies between 0 and 1, where 0 means the fragment was found in each protein of the bank, and 1 means no homolog was found in the bank. So let's jump directly into the tool and explore it point by point. So this is the tool. Let's read what they have written about it. They say that the surface identifies sites on the protein that are rare within the searched collection, i.e. to answer the question, what makes the protein unique among its homologs? So this is the rule of the tool. You have to give it the input structure, and you have to specify for it where to search for that structure. So let's see how we're going to do that now. First, it tells me the demonstration mode. If you want to try an example set, an example BDB structure, they have that here. So just run the demonstration mode. But since we're going to work on another BDB structure and go through the process by ourselves, so we will turn that off. So first step here tells you the BDB input. If you're not familiar with BDB yet, don't worry, we make a specific tutorial about that. So let's go to the BDB, and I know I want to have the structure ID called 1 U and D, and let's try BDB. That is it. I want to open that one. If you're not familiar with that, as I told you, don't panic. We will make another tutorial dedicated for the BDB. And now this is the structure of CDK5 with its activator P25, and it's bound by those inhibitors. So here I'm going to check how many chains do I have. I have here the cell division protein kinase 5. It's the CDK5. There are 2 chains, A and B, and now there's also the cyclin dependent kinase 5 activator one. This is its activator, and there are 2 chains for that, the C and D chains. And also I have a ligand, it's called the ALH, and it's in chains E and F. And for now, let's choose chain A from the structure called 1UNG. So let's go here and choose the database option and choose which database. We'll choose the PDB and write the code that we want, Y1UNG. I just will pick the chain A as I told you, and again let's press here press select. Let's go down here, and here it tells you the search space. What is the search space? As we told you, if you remember that, I told you before, we have to have a query structure, a query protein fragment or structure, and we have to determine the search space. Where is the tool going to look for your structure? So this is what we call the search space, as we said, and here it tells you they were going to search in the SCOP E. What is SCOP E? If you can see here, there is a question mark. We can press on that to know what we can enter here. And they tell you that this text area is only used to specify the SCOP subset to consider. So you can specify either all or any of the valid class or superfamily or family using the legacy format. Example, A for alpha helicoprotein, C.1.1.1 for triose, phosphate, isomerase family, or a list of codes is available. What does that mean? It looks complicated a little bit, but don't worry, we will simplify it now. So first, when I found something that I don't know, I Google it simply. So let's try Googling the SCOP E database. So let's try SCOP E and let's add the word protein to know what we're doing here. And let's search for it. And I found it. It's called Structural Classification of Proteins Extended. Let's press on that and know what is that. It's a database developed at the Berkeley Lab and UC Berkeley to extend the development of the SCOP database. There are 12 classes here, and they're numbered in alphabeticals A, B, C, D, E, F, and it's a good idea to know what family does your protein belong to. Whatever protein you want to know, its family or superfamily or whatever, its classification according to the SCOP database, you simply can write it here. So I'm going to write here CDK5, and let's look what is going to happen now. Actually, this is what came after we looked for CDK5. We can find here, this is the CDK5 activated one. This is the P25 that I told you about, that we're not interested in that tutorial. This is what we were interested in. This is the cyclin dependent PK CDK5. This is our protein. This is its classification code in this database, and we can click on that. This is family D.144.1.7 protein kinases catalytic subunit. This is the classification, and those are the proteins. We can you can see here that there are 66 proteins, as we will see, and those are the proteins belonging to this family. Now let's go back into our tool and see. So you may ask it to search in this family, or we can search the whole database and exclude that family, which logically will have more similar proteins. Or you may just, as we're going to do that now, I want to look in all the database. I'm not going to exclude anything. Here you can, as I told you can specify it in that format. You know, if you want to exclude a specific family or class, you can simply write it here, but I'm not going to exclude anything now for the sake of this tutorial. What is that? The k-mer length. You can just press here. It tells you the whole protein is scanned using a sliding window. This defines the window or k-mer size. You can find more details about that in the paper if you're interested. These are specific technicalities, and so I'm just going to leave it on the default. And here it asks you to specify the percentage identity that you want your protein. Let's read what they write here. They tell you that this field specifies the maximal sequence identity of the search protein collection. So leave it on the default too. Filter results, we're not going to apply any filter for the sake of that tutorial. And here is the last two parameters, the score constraints. It tells you the minimal BC score. This is the default, 0.95, and it tells you the BC score varies from -1 to 1. -1 means mirror, 0 means unrelated, and 1 means is identical conformations. So matches with BC scores values above 0.85 can be considered as significant, but can be largely distorted. Very similar and quasi-identical matches. So I'm going to leave it on default. And maximum rigidity, again, this is something too technical. If you want to know more about it, you can refer to the paper. And so for now, I'm going to leave it on default, and I'm going to run. Now it's running, as you can see, and it tells you there are 160 jobs already in the queue. Please wait. We're going to wait a little bit. Now it has finished. Those are the results. The first thing that you can see here is a cartoon representation where it's colored by the specificity score with the threshold. They tell you that most specific sites are displayed in red, the least specific sites are displayed in blue, and sites at specificity threshold are displayed in white. Increase this threshold a little bit and see what is going to happen. As you can see here, the blue are more than the red. The red sites, which are most specific sites, you can see our terminals and our loops here. So can you see that this can be targeted? I think it's so difficult to target something like that, or to find a pocket in here or here or here at the terminal. So, and most of the work will be done here, actually, in that area, in the blue sides, where there is the actual ATP binding site, because this is a kinase actually. So, in our case, if I want to inhibit the CDK5 because it's involved in Alzheimer's disease. So if I inhibited that, if I went to just designing something that binds into a pocket in these blue regions, say the ATP binding pocket or ATP binding site, I'd be ending up having lots of off-target effects, off-target interactions and off-target side effects and so on. So this will not be a good choice for me. But do you think that this is always the case, that you always want to have more of a red color in your structure result here and less of the blue ones because you want it to be more specific and unique? Please tell me in the comment what you think, but in my opinion, the answer is not. This is not correct all the time. The answer is it depends. It depends on the products you want to do and the design that you have in mind. Let me explain that for you. For example, if you want to design a compound that acts on multi-target, for example, if you're working on Alzheimer's disease and we want to target the CDK5 and we want to target the acetylcholinesterase, in that case, it may be useful to define the SCOP family of the acetylcholinesterase and tell the tool to look in that search space. So it will give you the similar size, and in that case you will be looking at the blue regions, and you will be so lucky if you found a blue region in both proteins that form a druggable pocket. What do I mean by druggable pocket? That will be a topic of a next tutorial. Stay tuned for that. Hit the subscribe button of this channel and of our newsletter to stay informed when we release it. But for now, let's imagine that we found a pocket in here and a pocket in here, so we're going to look for the blue regions, which are least specific or similar. Okay, so how can we know that? We can do a beautiful thing here. You can download all the results you have here. These are the residue number or index. And this is the specificity score of each residue. And the nice thing about that is that you can download here all the results. You will have a CSV file that you can edit in Excel, having these data where you can sort and set a threshold. In that case, if you're looking to find similar regions, you will be looking for those sites with the specificity score closer to 0. In the other case where I'm just designing something that I want it to be specific, so I will be looking for those residues or forming sites with the specificity score closer to 1. The closer to 1, the better. Okay, the closer to 1, that means they are different and they are very specific and they are unique to that protein, as they said. So you can can note the number of those residues and visualize them in your visualizer, and you can then check if they form a druggable pocket. You will be done knowing if the site you're targeting is specific to that protein or not. And as we said, it's according to the context of your research project and idea of design. And that's it for now. I hope that this tool added some insights to you and that this was an informative tutorial for you. Please let me know in the comments what you think, and if you can think of other design ideas that you can apply this BCSpecificitySearch tool to. And don't forget to hit the subscribe button of this channel and subscribe to our newsletter to get informed about our courses and our tutorials when they are released. Thank you so much and see you soon.